Good morning, students. Now we shall learn the health and diseases lesson part 3. In the previous video we have learned about the immune system up to lymphocytes. Now we are going to learn what are these antigens. The term antigen is used in two senses. The first one is to describe a molecule which will generate an immune response in the body. Secondly, it is considered as a molecule that will react with the antibodies. Antigens are large complex molecular substances that can induce an immune response. Antigen is a substance that is specific to an antibody or T receptor cell, and it is often known as immunogen. Now we have to learn the definitions of an immunogen: haptens, epitope, paratope, and adjuvants. What is an immunogen? It is a substance which is capable of initiating an immune response. Haptens are non-immunogenic substances that can react with the products of the immune response. Epitope is the active part of an antigen that will bind with the antibody, and paratope is the part of the antibody that can recognize and binds with an antigen. So epitope is present in the antigen and paratope is present in the antibody. They both only will join with each other in the antigen-antibody reaction. What are these adjuvants? Adjuvants are the substances that can enhance the immune response of an antigen. And these adjuvants are used in the pharmacology, which is considered as an important agent that improves the immune response of a vaccine. What are the types of antigens? On the basis of the origin, antigens are classified into exogenous antigen and endogenous antigen. The antigens which enter the host from outside in the form of microorganisms, pollens, drugs or pollutants are called as exogenous antigens. And the antigens which are formed within the individual's body are termed as endogenous antigens. The best examples for the endogenous antigens are the blood group antigen. Now let us see what are these immunoglobulins. Immunoglobulins are nothing but the antibodies. They are the protein molecules synthesized on exposure to the antigen by the B cells, and this can combine specifically with the antigen. When the pathogen enters, the B lymphocytes will produce an army of plasma cells that can produce a protein called antibodies to fight with them. So antigen is represented by Ag and antibody is represented by Ab. The antibodies are classified into 5 major categories. They are IgG, immunoglobulin gamma, IgM, immunoglobulin mu, IgA, immunoglobulin alpha, IgD, immunoglobulin delta, and IgE, immunoglobulin epsilon. Let us learn the structure of immunoglobulin. The basic structure of an immunoglobulin was revealed by Porter and Edelman in 1950. An antibody molecule is a Y-shaped structure, and it comprises of 4 polypeptide chains, 2 identical light chain and 2 identical heavy chains. The light chain is represented by L and the heavy chains are represented by H. The molecular weight of light chain is 25,000 Dalton and it has approximately 214 amino acids, whereas the heavy chain has the molecular weight of 50,000 Dalton and has 450 amino acids. The polypeptide chains are linked together by disulfide bonds, SS bond. One light chain is attached to each of the heavy chain, and 2 heavy chains are attached to each other in the form of a Y-shaped structure. Hence, an antibody molecule is represented by H2 and L2. The The heavy chain is flexible at a region called hinge region that lies approximately in the middle. The L chain and H chain has two terminals. One is called the C terminal or carboxyl terminal, and another one is the amino or N terminals. And each chain has two regions, namely variable region, which is represented by the letter B, and the large constant region, which is represented by the letter C. Antibodies that are responding to different antigens will have different variable regions, but they have the same constant region. In each arm of the monomer antibody, the V regions of the heavy and the light chain, the variable region of the heavy and the light chain will form the antigen binding site. And this is the antigen binding site, and it is shaped to fit a specific antigen. And consequently, each antibody will have two antigen binding regions. This is 1 and this is 2. The constant region forms the stem of the antibody, and this constant region will determine the antibody class. The functions of the immunoglobulins are agglutination, precipitation, opsonization, and neutralization. Antigen-antibody interaction. How do they react with each other? The reaction between the antigen-antibody forms the basis for the humoral immunity or antibody-mediated immunity. The reaction between the antibody and antigen takes place in 3 stages. During the first stage, it forms the antigen-antibody complex. In the second stage, visible events like precipitation and agglutination will take place. In the final stage, it causes the destruction of the antigen or its neutralization take place. The binding force between the antigen and antibody is due to 3 factors. They are the closeness between the antigen and antibody, second one non-covalent bonds or the intermolecular forces, third one affinity of the antibody. When the antigen and antibody are closely fitted, their strength of binding is great, and when they are apart, the strength is very low. The bond that holds the antigen to the antibody is the non-covalent bond in nature, and these bonds include hydrogen bond, electrostatic bonds, van der Waals forces, and hydrophobic bonds. About all these bonds you would have learned in chemistry lessons. Antibody affinity is the strength that lies between a single antigenic determinant and a single antibody. Applications of antigen-antibody reactions. First one helps to determine the blood group for transfusion. It helps to study the serological ascertainment of exposure of infectious agents, like whether the person is exposed to COVID-19 virus or not can be identified by examining their serum, blood serum. When it is examined for the particular antibody, it can be confirmed whether the person is having COVID-19 infection or not. It helps to develop immunoassay of various substances. It helps to detect the presence and the absence of the protein in the serum, a particular protein in the serum, and also helps to determine the characteristic feature of the immunodeficiency diseases. Let us see the different types of antigen-antibody reaction. First one is called precipitation. This is a reaction between the soluble antigen with the antibody, and which will form visible precipitate. This reaction is called precipitin reaction, and the antibody that brings about this precipitation is called as precipitin. Whenever a particulate antigen interacts with the antibody, it results in the clumping or agglutination of that antigen, and this is called as agglutination reaction. The antibody that brings about the agglutination is called as agglutinin. The third interaction is called opsonization or enhanced attachment. In this process, the binding of an opsonin or the antibody take place. It attaches, this opsonin molecules will attach itself to the receptors of the pathogen cell membrane. After the opsonin bind to the cell membrane, This will enhance the phagocytosis of the pathogen. The phagocytes will be attracted to the pathogen and it causes phagocytosis. So, opsonization is a process in which the pathogens are coated with the substance called opsonin, that is the marking the pathogen for the destruction. That is the marking of the pathogen for destruction by the immune system. And this results in a much more efficient phagocytosis. The fourth reaction is called the neutralization reaction, which involves the elimination of the harmful bacterial exotoxins or a virus by specific antibodies. Here the neutralizing substances are the antibodies and they are known as antitoxins, and they are produced by the host cells in response to a bacterial exotoxin, and these substances are called antitoxins to inactivate the toxins. They will neutralize the exotoxins and the virus. What are vaccines? Vaccine is a biological preparation which helps to provide acquired immunity to a particular disease. It resembles the disease causing microorganism, but it will be in the weakened or attenuated or killed forms of microbes, or their toxins, or one of its surface proteins also can be introduced into the person's body to trigger or to initiate active immune system. The vaccines will teach the body how to defend itself from the disease causing pathogens. Vaccines will give only very little amount of the weakened viruses or bacteria or even part of them, but this will help the immune system to recognize the organism without actually causing the disease in the body. Sometimes these vaccines need to be given more than one time, that is called as booster vaccination, and this will make sure the immune system to overcome the infection in the future. Vaccines initiate immunization process. Vaccines are classified into first, second and third generation vaccine. First generation vaccine is again divided into live attenuated vaccine, killed vaccine and toxoids. In the live attenuated vaccines, they use the weakened, aged or less virulent form of the viruses. Example for these vaccines: measles, mumps, rubella, MMR vaccine, varicella vaccine, chickenpox vaccine. Secondly, killed vaccines. These are prepared by killing the microorganisms which are causing the diseases by heat or any other method. Example for this is sac polio vaccine. Thirdly, toxoid vaccines, and these vaccines will contain the toxins or the chemicals which are secreted by the disease causing bacteria or virus, and these vaccines will help us to fight against the harmful effect of the infection rather than the organism itself, rather than the microorganism which causes the disease, that is instead of infection itself. Example for this is DPT vaccine, which will help us to fight against diphtheria, pertussis and tetanus. Now second generation vaccines. Second generation vaccines contains purely the surface antigen of the pathogen. So all the pathogen will have the surface antigens on their cell surface, and they will be separated and they are introduced into the person's body to trigger their active immunity. Instead of injecting the whole organism, they take the surface antigen alone, and this alone will be injected into the person to trigger or to induce the active acquired immunity. Example for this is hepatitis B vaccine. The third generation vaccines are purest and they are having high potential to fight against diseases, and they are synthesized artificially. The latest revolution in vaccine is the DNA vaccine or recombinant vaccine. So DNA vaccine and recombinant vaccines are the present day vaccines. Now here you can see how the vaccines are of different types. In the live vaccine, the natural microorganism will be administered into the person's body in a very small amount, so that it will not cause the disease. Instead, it will trigger the active immunity in their body. In the live attenuated vaccine, the live pathogens will be weakened by heat killing or any other method. Thirdly, the live pathogen will be inactivated, or some particles of the microbes will be injected into the person's body to initiate their active immunity. Fourthly, the toxins which are secreted by the pathogens are injected into the person's body as vaccines, and they will help us to produce the active immunity in our body to fight against the actual infection. Example for this is tetanus toxoid. Dr. Edward Jenner prepared the first vaccine for the smallpox in 1796, and polio vaccine was developed by Dr. Jonas Salk, and so it is called Salk polio vaccine. Dr. Albert Sabin produced the first live attenuated oral polio vaccine, and Louis Pasteur in 1885 discovered the vaccines for rabies, anthrax and cholera. BCG vaccine was developed by Colmetti and Guarin against tuberculosis. So B for bacillus. The tuberculosis is caused by a bacillus bacteria and found by Colmetti and Guarin in 1908. Now, how to differentiate between vaccination and immunization? Vaccination is the process by which the vaccines are administered into the body. This induces the body to produce an immunity to a specific disease, whereas immunization is the process of building up of the immunity. This describes the actual changes that happens in the body after receiving a vaccine. So vaccine works for fighting the pathogen, and it records it in the memory system as memory cells. So when the same pathogen enters the body for the next time, it gives a quick response and eliminate the pathogen out. The body is able to fight against the disease, and that is called as immunity, and we can say the body being immunized against the disease. The hypersensitivity is the overactive immune response or exaggerated immune response to certain antigen present in the environment, and it is called as allergy. And the substance which causes this allergy are called as allergens. An allergen is an antigen that causes the allergic reaction. The common examples of allergens are mites in the dust, pollen, and some proteins in the insect venom. Hay fever and asthma are some examples of allergy. The symptoms of allergic reaction include the sneezing, watery eyes, running nose, difficulty in breathing, etc. Allergy is a form of overactive immune response, and that is caused by IgE immunoglobulin and mast cells. And it can also be due to the release of certain chemicals like histamine and serotonin from the mast cells. Anaphylaxis is a term which is given for the sudden, systematic, severe, immediate hypersensitivity reaction, and it occurs as a result of rapid generalization of the mast cell degranulation. Anaphylaxis is the hypersensitivity reaction. Next we have immunodeficiency diseases, which causes ineffective immune response. The primary immune deficiencies are caused by genetic developmental defects, but the secondary immune deficiencies are due to various reasons like radiation, use of cytolytic or immunosuppressive drugs, use of certain chemicals, medicines, and infection. For infection, there is one example, that is AIDS. AIDS is acquired immunodeficiency syndrome. It causes deficiency of immune system, and this is acquired during the lifetime of an individual. And so it is not congenital disease. It is caused by the infection of a virus called human immunodeficiency virus, and this virus will selectively infect the helper T cells and destroy them, by which the helper T cells cannot stimulate the antibody production by the B cells, that eventually results in the loss of natural defense mechanism in the body. This HIV can be classified into HIV-1 and HIV-2 types. Structure of HIV. HIV belongs to the genus lentivirus, and when it is observed under the electron microscope, it is a spherical virus with 100 to 120 nanometer diameter. It contains the dense core which is surrounded by a lipoprotein envelope, and this envelope has glycoprotein spikes, and they are termed as glycoprotein 41 and glycoprotein 120. So these vertical ones are glycoprotein 41 and the horizontal ones are the glycoprotein 120. At the core region it has two large single-stranded RNA, and this is attached to the molecules of the reverse transcriptase. It contains the enzymes called protease and ribonuclease. The core is covered by a capsid which is made up of protein, and another layer of matrix protein is also seen. HIV is often located in the macrophage cells of the body. It can survive for about 1.5 days inside the cell, but outside it can survive only for 6 hours. How does it get transmitted? HIV transmission takes place through unsafe sexual contact, blood contaminated needles, organ transplantation, blood transfusion, and vertical transmission from the mother to the child. But HIV will not be transmitted by the insect bite or the casual contacts. Once the HIV virus got into the body, it enters into the macrophage cells. And here the RNA genome of the virus will replicate to form the viral DNA with the help of the enzyme called reverse transcriptase. And this viral DNA will get incorporated with the DNA of the host cell, and it will direct the cell. The viral DNA will direct the cell, infected cell, to produce the viral particles. The macrophage will continue to produce a virus, and in this way it becomes a HIV factory. Simultaneously, this HIV which are coming out of this macrophages will enter into the T lymphocytes, and there they replicate and produce more progenies of the virus. And these viruses will attack the other cells of the blood, other helper T lymphocytes, and this is repeated, leading to a progressive decrease in the number of the T helper cells in the body of the infected person. At this time, the person will suffer from fever, diarrhea, and weight loss. And the person who is suffering from the infection becomes immune deficient, and he or she will be unable to protect against any infection. How to identify the HIV infection? A simple blood test is available to determine whether the person is infected or not. That is called ELISA test, or enzyme linked immunosorbent assay, and this will detect the presence of HIV antibodies in the serum. It is only a preliminary test. Western blot test is more reliable, and it becomes a confirmatory test. If both the tests confirms the presence of the viral core proteins, then the person is considered as a HIV positive. AIDS has no cure, so prevention is the only option. Advocating safe sex, promoting regular checkup, safe blood transfusion, use of the disposable needles, use of the condoms during sexual contact, prevention of the drug abuse, AIDS awareness program by NACO, NGOs, WHO will help to prevent the spread of the disease AIDS. Next we have autoimmune disease. Autoimmunity is an abnormal immune response in which the immune system fails to distinguish the self and the non-self molecules, and it will attack its own body molecules. So autoimmunity is a misdirected immune response. Here our body produces antibodies that are autoantibodies and cytotoxic T cells to destroy our own tissue. And this diseased state is called as autoimmune disease. When the cells act as the antigens in the same body, they are called as autoantigens. And the antibodies which are produced for this autoantigens are called autoantibodies. These autoimmune diseases can be divided into two broad categories, like organ-specific and non-organ specific autoimmune diseases. In organ-specific, it is directed, the immune response is directed mostly to a particular organ, mostly against a particular organ, and the autoantibodies will block the functions of that particular organ. Example for this is Hashimoto thyroiditis, Graves' disease of thyroid gland, and Addison disease of adrenal gland. In non-organ specific disorder, the autoimmunity is widely spread throughout the body. Example for this is rheumatoid arthritis and multiple sclerosis. Tumor immunology. A tumor or neoplasm is a group of cells whose growth has gone unchecked. The tumor will continue to grow and invade the healthy tissues. It is called as cancer. When it spread to the other parts of the body and give rise to the secondary tumor, then it is known as metastasis. And this tumor may be benign or malignant. When it is benign, it is non-cancerous tissue and it is capable of indefinite growth and do not invade the other parts. Whereas in the malignant tumor, the cells will grow indefinitely. It will get detached from the main part and it will migrate to the healthy surrounding tissue and invade them. In normal cell, the cell growth and the differentiation is highly controlled and regulated. Whereas in the cancer cells, it is not done. There is a breakdown of this regulatory system. Normal cells will show a property called contact inhibition, which will inhibit the uncontrolled growth. But in the cancer cells, this property is not present. As a result, these cells will divide continuously, giving rise to mass of tissue called as tumors. The concept of immunological surveillance, which means the primary function of our immune system is to check thoroughly throughout our body to seek and destroy this malignant cell that are caused by the somatic mutation. If this surveillance mechanism's efficiency reduces, it causes cancer. And this happens as a result of aging or due to any congenital or acquired immunodeficiencies. So, if the immunological surveillance is effective, cancer will not be caused. They will not get cancer. But in case if it is suppressed, if there is a lapse in this surveillance, then it causes tumor. When a cell undergoes malignant transformation, and a particular cell undergoes a transformation as malignant cell, it will acquire new surface antigens on its cell membrane. By losing some normal antigens, it will acquire some new surface antigens. And these antigens, when they are present on the membrane, will be identified by the immune system and they induce an immune response. And this immune response is given by both humoral as well as the cell-mediated immune system. It can be both humoral or cell-mediated immune response. Cancer cells sometimes avoid this immune detection because they are not the foreign bodies, but they are the abnormally functioning body cells. And this makes them difficult to treat. Immunotherapy of cancer. Immunotherapy of cancer involves the use of the monoclonal antibodies. And these antibodies are produced either by the body or in the laboratory. Different approaches have been attempted in the treatment of the cancer. Immunotherapy appears to be very important to get rid of the residual malignant cells. Even if the gross tumor lumps are removed out of our body, the residual malignant cells which are present in the body can cause cancer. And this can be avoided. This could be removed. This could be got rid with the help of the immunotherapy. The best results in the treatment of the cancer could be attained by following an integrated approach. Integrated approach as a combination of surgery, radiotherapy, chemotherapy and immunotherapy. Now, what are the differences between the normal cell and the cancer cell? In this picture itself we can see the normal cells are small and they have uniformly shaped nucleus. And the cytoplasmic volume is relatively larger than the nucleus, whereas in the cancer cell the nucleus is larger and it is variably shaped and it has small cytoplasmic volume related to the nucleus. The cell size, shape and cell arrangement in the tissue of the normal cells are uniform and proper, whereas there is variation in the cell size, shape and the organization of the arrangement of the cells are also not proper. Normal cells may possess differentiated cell structure and they have normal presentation of the cell surface marker. Whereas in the cancer cells there will be loss of normal specialized feature, and they have some elevated expression on the surface of the cell. Elevated expression of the certain cell marker could be clearly seen on the surface like crab-like appearance, and that's why this disease got the name cancer. Here the normal cells divide slowly and they have very well defined cell boundaries, and cell tissues are clearly demarcated. But the cancer cells will divide at a faster rate and their boundaries are poorly defined. Their cell tissues will have poorly defined tumor boundaries. The cell boundary and the tissue boundaries are clear in the normal cells, whereas it is not so in the tumor cells. Adolescence begins at the age of 12 years with a rapid physical and sexual development called puberty up to maturity at the age of 19 years. During this period, the individuals will have lot of psychological and social changes. They are more prone and vulnerable to peer group. They are pushed to experiment with drugs and alcohol. During this age, proper education and guidance must be given, and this will help the youth to say no to drugs. Alcohol, what is it? It is a psychoactive drugs which will act on the brain of a person and affects their mind and behavior. It also acts as a depressant which will slow down the activity of the nervous system. The intake of certain drugs frequently impairs one's physiological and psychological functions is called as drug abuse. The commonly abused drugs are opioid, cannabinoids, cocoa alkaloids, barbiturates, amphetamines and LSD. LSD for lysergic acid diethylamide. What are these opioids? Opioids are the drugs that binds to the specific opioid receptors of our central nervous system and intestinal tract. Heroin is a crystalline compound and it is chemically diacetyl morphine. It is white in color, odorless and bitter in taste. It is obtained by the acetylation of morphine, and it is extracted from the flowers of the poppy plant. This is the poppy plant. Morphine. Morphine is one of the strongest painkiller and it is used during the surgery. It is the most widely abused narcotic drugs. It acts as a depressant and it will slow down the body functions. Cannabinoids are a group of chemicals which are obtained from the cannabis sativa, which is commonly called as Indian hemp. This picture shows the Indian hemp plant. Natural cannabinoids are the main source of the illegal drugs like marijuana, ganja, hashish and charas. These will interfere with the neurotransmitter dopamine in our body. The cannabinoids have a potent stimulating action on the central nervous system and produces increased energy and a sense of euphoria. Euphoria means feeling high of oneself. It is an extreme unrealistic feeling of physical and emotional well-being. Cocaine. Cocaine is a white powder which is obtained from the leaves of a coca plant, Erythroxylum coca. It is commonly called as coke or crack. Cocaine causes serious physical, psychological problems including hallucination and paranoia. They both are like delusion or exaggerated self-importance. Atropa, belladonna and datura are the other plants which have hallucinogenic properties. Drugs like methamphetamine, amphetamines, barbiturate, tranquilizer, lysergic acid diethylamide, they are normally used to treat the patients with mental illness like depression and insomnia, but they are often abused. Tobacco. Tobacco is smoked in the cigarettes, chewed as the tobacco leaves, used as snuff powder, mukupodi. Use of this will increase the carbon monoxide content in the blood, causing oxygen deficiency in the body. Tobacco contains nicotine, carbon monoxide, tar, which cause problems in the heart, lungs and the nervous system. Adrenal glands are stimulated by nicotine and they release adrenaline and noradrenaline, which increases the blood pressure and the heartbeat. These are some of the drugs which are used as stimulant, depressant, analgesic, cannabis and hallucinogens. Stimulants. Drugs like amphetamines, cocaine, nicotine and tobacco are used as stimulants. In common they say ஊக்க மருந்து, which they use illegally before sports performance. They use this. This accelerates the activity of the brain. Stimulants accelerates the activity of the brain. Depressants. Drugs like alcohol, barbiturates, tranquilizers are depressants and they will slow down the activity of the brain. Opium and morphine are used as narcotic or analgesic. They act as a depressant of the central nervous system. They are legally used as painkillers. Cannabis, bhang, marijuana, ganja and charas are the drugs which are cannabis, which will affect the cardiovascular system. Hallucinogens. Lysergic acid diethylamide, that is LSD. Phencyclidine. These are the drugs which are used as hallucinogens and they will distort the way one sees, hears and feels. They will change the way one sees, hears and feels. Addiction and dependence. Addiction is a physical, psychological need to use the substance like alcohol, etc. Addiction is harmful to the individual. The addictive behavior of a person is destructive. This also leads the individual to lose their jobs, homes, money, friendship, family, relationship, etc. Addiction to the drugs and alcohol can lead to the effects like euphoria or temporary feeling of well-being. Repeated use of this alcohol and drugs will affect the tolerance level of the receptors in the body, and this makes them to take high dose of the drugs and the alcohol, leading to the excessive destruction, and it leads to physical and psychological dependence on the drugs and the alcohol. The psychological dependence, we call it as hooked on. The user gets mentally hooked on to the drug. The drug users constantly think about only the drugs. They will not have any other thought and they become uncontrollable, craving for it all the time, and this state is called euphoria. That is, addicted person will become mentally, emotionally preoccupied with the drug alone. Physical dependence is a state in which the user body requires the continuous presence of the drug in their body. If they stop taking it abruptly, it will lead to some withdrawal symptoms. In a sense, the body becomes confused and it will protest against the absence of the drug. They will have range of symptoms like a mild tremor or convulsions like nadukam, severe agitation. They become agitated and fits comes. They get depressed mood, anxiety, nervousness, restlessness, irritability, insomnia, dryness of throat, etc. depending on the type of the drug they abuse. What are the effects of drugs and alcohol? The short-term effects are euphoria, pain, dullness of sense, alteration in behavior, blood pressure, narcosis, that is deep sleep, nausea and vomiting. And the long-term effects are the serious damages to the person, both physical and mental disturbance take place and the user gets unbearable tortures. For example, heavy drinking permanently will damage the liver and the brain. Use of alcohol during adolescence may have long-term effects. They will interfere with the liver function. Fat accumulation in the liver cells causes scar tissues, scarring of the liver called liver cirrhosis. Alcohol irritates the stomach, causes ulcer. It leads to the risk like high blood pressure, stroke, coronary artery disease, heart attack, etc. Korsakoff syndrome. What is it? It is a chronic memory disorder. It is caused due to alcohol misuse. What is alcoholism? Alcoholism is the inability to control the drinking. due to physical and emotional dependence on alcohol. Prevention and control is practically possible to prevent someone from using drugs and alcohols. Here are some ways. First one, effectively dealing with the peer pressure. The biggest reason for the teens to start with the drugs is due to their friends and peer groups who will impose them to do so. For this, one must be careful enough to choose better friends. They must choose best friends to avoid such bad habits. Secondly, seek the help from the parents and peers. Sometimes parents and the close and trusted friends, they will help them to sort out the problem and come out of their anxiety and guilt instead of getting into harmful habits. Thirdly, education and counseling. This creates a positive attitude to deal with the problems and accept the disappointments in life by the youth. Fourthly, looking for danger sign. Teachers and parents should look for the sign that indicates the tendency of the youth to go in for addiction. So they have to be careful enough to find out the vulnerable person and help them to come out of it. Seeking professional and medical assistance. This is available in the form of highly qualified psychologists, psychiatrists, de-addiction and rehabilitation programs to overcome their problems. Mental health is the state of well-being of mind with self-esteem. What is this self-esteem? It means liking yourself, one's own self, and this will help them to stand up for what they believe is right. Positive mental health is very important for wellness. A mentally healthy person will reflect a good personality. Mental health also improves the quality of life, and they will cooperate with others and will be appreciated and rewarded by the society. Depression is a common mental disorder. This causes people to experience depressed mood, loss of interest or pleasure, feeling of guilt or low self-worth, disturbed sleep and poor appetite, etc. What are the signs and symptoms of mental depression? They are loss of self-confidence and self-esteem, anxiety, not being able to enjoy things which are usually pleasurable and interesting, change of lifestyle like exercise, meditation, yoga, healthy food habits can help one to get rid of the depression. Exercise will stimulate the body to produce the serotonin and endorphin, which are the neurotransmitters which helps to suppress the depression. Practicing exercise in a daily life will create the positive attitude towards life. Participating in an exercise program will increase the self-esteem, boost self-confidence, create a sense of empowerment and enhance the social connection and relationship. Brain is one of the most metabolically active part of our body and it needs steady stream of nutrition to function. Poor diet may not provide sufficient nutrients and that may also provoke the symptoms of anxiety and depression.